Hello and welcome to my hello and welcome to the Walking Cell, and thank you for continuing to explore life together. In chapter 5, we discussed the walking cell. So here we actually experience how dynamic the living cells are. So when we studied the structure of cells, we said that the cells are separated from their surrounding environment by the plasma membrane. And the plasma membrane is made of phospholipid bilayer and protein molecules that are sandwiched between it. So the plasma membrane and its proteins actually enable the cell to survive and to function, creating a compartment in which the cell can have regulation over, so that you can have portions inside the cell that have a different concentration of molecules than the surrounding. In order for the cell to be functional, to be the walking cell as described, the membranes must be able to isolate the cell from its environment, and different parts of the interior of the cell also compartmentized for functions to be made possible. In order for the cell to work, energy and matter must be available. So the cell must synthesize the membranes themselves and also utilize energy in that process, and all of the dynamics of working, moving materials, making body parts utilize energy. And so the cell has energy transformation processes that's internal to the cell. The combined chemical reactions that take place in the cell is known as cellular metabolism. This process is metabolic processes are enhanced and made efficient thanks to the enzymes. So the big picture for this chapter will discuss membranes, energy, enzymes. These contribute to the working cell. So this slide just shows you a model describing what the biological membranes look like that are so essential to the functioning and survival of a living cell. As discussed in previous chapter, they are made of phospholipid bilayer. So these yellow phosphate heads and the green fatty acid or fatty acid tails, spontaneously respond in an aquatic environment. These red and gray balls representing the aquatic environment inside the cell and outside the cell. So spontaneously, the phospholipids arrange into a bilayer of phospholipid, one layer, and then the second layer facing it. Notice also the presence of water molecules, illustrating the ability of water to move across, facilitated by these blue aquaporins. And so in this chapter, we're going to discuss how the membrane works, the construction or the structure of the membrane, the cell membrane or biological membranes illustrate how throughout the entire world of biology, structure and function go hand in hand. In addition to the phospholipid bilayer, there are proteins that are sandwiched in between the phospholipid bilayer, and they are not rigid. They can move around as a fluid bilayer, this oily fatty acid parts, fluid. And so this allows the cell membrane to be very, very dynamic and functional. So in this chapter, the big ideas, we look at the membrane structure and function. Remember, the entire theme in biological systems is that the structure enhances or determines the functions of an organelle or a cell or a system. So we're going to look at the energy and the cell. Every living cell undergoes the process of cellular respiration. All other forms of energy will be converted to ATP, the energy currency of the cell, during the process of cellular respiration. This is the molecular form of energy that all cells can utilize to do work. The process of the metabolic processes that take place to make the cell function overall is made effective by the help of enzymes, chemicals that speed up the rate of chemical reactions in the body, such that an experiment that maybe would have taken a month to complete will take place in seconds. And several reactions will take place in seconds in the cell, thanks to the enzymes. Now we're taking a look at the membrane structure and function. So the accepted model that biologists use to describe the biological membrane or the cell membrane is the fluid mosaic model. The fluid referring to the fatty acid tails, which has about the same consistency as your fluid olive oil. And mosaic because of the diverse protein molecules that are suspended in this fluid phospholipid bilayer. Important property of the plasma membrane is that they are selectively permeable, meaning that they allow certain substances to pass through it, and they allow selected substances to pass through it at different rates, and allow some substances to pass through, and some will not be allowed to pass through at all. So they are selectively permeable. And the proteins that are suspended in the phospholipid bilayer, they are diverse, and they are diverse in form, and they perform various functions in the cell. So this illustrates the functions of the plasma membrane. So this is illustrating two adjacent cells. This is the cell membrane of one cell, and this is the cell membrane of the next cell. So looking at the cell of a multicellular organism. So this is the phosphate head, fatty acid tail, phosphate head, fatty acid tail. So this is one layer, this is second layer, and so we have 1, 2 bilayer of phospholipid. From the extracellular side of the membrane, extracellular means outside of the cell, oxygen can easily diffuse across. So these non-polar molecules, small non-polar molecules such as oxygen and carbon dioxide, can diffuse across the biological membrane. Oxygen that's so much needed inside the cell can simply diffuse across the membrane. And once cellular respiration takes place, a byproduct of cellular respiration is carbon dioxide. The body does not want to accumulate carbon dioxide and become an acidic environment. Luckily, carbon dioxide can diffuse across the biological membrane back into the extracellular matrix, through which it can then go out of the system or the body or the organ. So the membrane proteins that are suspended in the phospholipid bilayer, they can act as enzymes to speed up the rate of chemical reaction. So this is, for example, a reactant. This is enzyme number 1. It helps to change it to an intermediate product. And then the enzyme number 2 helps to change it to the final product. So you can have a cascade of enzymes along the surface of the membrane, helping a series of metabolic reactions to take place until the final product is produced. In animal cells, cholesterol is also sandwiched between the phospholipid bilayer. Cholesterol, they're very, very important in making sure that the phospholipid bilayer is not too closely and tightly packed that it cannot function in transport and metabolism. So it enhances the fluidity and the flexibility and the versatility of the biological membrane. Other membrane proteins could be glycoproteins that enables the cells to recognize the neighboring adjacent cells. So it can recognize cell, or it can recognize foreign unwanted cells. So in developing embryos, cell recognition membrane glycoproteins would help the cells to decide which ones will stick together to form specific organs, and so on and so forth. So those are membrane proteins. Membrane proteins also enhances the fusing together of cells in multicellular organisms. Some ions that are charged particles are not able to pass through the phospholipid bilayer. And so some of the proteins that are suspended in the layer would help to facilitate the diffusion of materials of these ions into the cell and facilitate the diffusion. Certain ions need to be moved out of the cell against the concentration gradient in active transport. So with the help of ATP, proteins would help to, or protein channels by active transport would help to move materials against the concentration gradient. The presence of chemicals can also be sensed. So we have a signal transduction in cells thanks to the proteins that are suspended in the membrane. They recognize a chemical signal, and then they form secondary messengers that relay that signal into the interior of the cell, causing the cell to give appropriate response to that stimulus from outside the cell. Membrane proteins also attach to the cytoskeleton and the cytoplasmic side, the interior of the cell, and then to the extracellular matrix fibers, helping to give integrity in multicellular organisms. So as you can see, there are several roles for the cell membrane. Again, let's quickly look at this. They allow diffusion of small molecules. They have protein suspended in the phospholipid bilayer that speed up the rate of chemical reaction acting as enzymes. Glycoproteins helps to recognize the other cells and molecules outside of the membrane. Junction proteins help cells in multicellular organisms to stick together. Suspended proteins channels such as the ATP synthase involved in active transport help to move materials against the concentration gradient. Channel transport proteins help to move materials passively across the phospholipid bilayer. Receptor proteins act in signal transduction across the membrane, and then attachment proteins help to solidly attach the cytoskeleton cell membrane to the extracellular matrix. So small nonpolar molecules diffuse readily across the phospholipid bilayer. The transport proteins allow specific ions or molecules to enter or exit the cell, either by passive transport across channel transport proteins or by active transport using ATP. The difference between passive transport and active transport. Passive transport is just movement down a concentration gradient from where there are more molecules to where there is less. Active transport is moving from where there is less to where there is more, involving the use of ATP. That distinguishes passive from active transport. Enzymes, some membrane proteins are enzymes, and they may be grouped to carry out sequential reactions that are essential for life and survival. Attachment proteins attached to the extracellular matrix and the cytoskeleton, helping to support the membrane and coordinate external and internal changes. Signaling molecules, they bind to receptor proteins. Receptor proteins relay messages by activating other molecules inside the cell, such as cyclic AMP and so on and so forth. Junction proteins help to form intercellular junctions. If you remember earlier on, we described tight junctions, gap junctions, and so on and so forth. Glycoproteins, they serve as ID tags, so they may be recognized by membrane proteins of all the cells. Now let's take a look at an overview of the cell signaling process.
Cells communicate with one another by means of chemical signals. For the receiving cell, there are 3 stages in the signaling process: reception, transduction, and cell response. The cell targeted by a particular signal has a receptor molecule complementary to the signal molecule or ligand. The ligand fits like a key in a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the receptor to a form that can bring about a cellular response. This might involve a series of steps, a signal transduction pathway that alters and amplifies the change. In the 3rd stage of cell signaling, the transduction process brings about a cellular response. This can be any of many different cellular activities, such as activation of a certain enzyme, rearrangement of the cytoskeleton, or activation of specific genes. Receptors that are plasma membrane proteins usually affect the cell through multi-step signal transduction pathways. These pathways allow for amplification of signals and signal coordination and regulation. Signal transduction often involves smaller molecules called second messengers and protein phosphorylation by enzymes called protein kinases. Second messengers are small, non-protein molecules that act as intermediaries in signal transmission. 2 important second messengers are cyclic AMP and calcium ions.
Cyclic AMP acts as a second messenger in pathways initiated by both G protein-linked receptors and receptor tyrosine kinases.
The hormone epinephrine acts via cyclic AMP as a second messenger. An epinephrine molecule docks with a protein, which acts through a G protein to activate the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP, or CAMP, which quickly diffuses through the cell and triggers further steps in the signaling pathway. In this cell, we show it activating a protein kinase. Calcium ions also act as second messengers in signal transduction pathways.
This time we will show a receptor tyrosine kinase, but G protein-linked receptors can also initiate release of calcium ions. The signal molecule docks with the receptor and activates an enzyme called phospholipase C. The enzyme cleaves a small molecule called inositol trisphosphate, IP3, from a certain kind of membrane phospholipid. IP3 docks with a calcium channel on the endoplasmic reticulum. The channel opens, releasing calcium ions into the cytoplasm, where they activate proteins that carry out cell responses.
Called protein kinases are important in many signal transduction pathways. Protein kinase catalyzes the transfer of phosphate groups from ATP to protein. often than not, the activated protein is also a protein kinase, which may act on still another protein kinase. One kinase may activate many molecules of the next type of kinase in the chain, thus amplifying the signal until the last kinase activates many molecules of the protein that carries out the final cellular response.
So think of the word kinase. So kinetic, ASE, all enzymes typically have their name suffix as ASE. So kinetic, dynamic, moving, functional proteins are kinases as they are amplified. So, from the evolutionary perspective, biologists believe that spontaneous formation of membranes was an initial critical step in the origin of life. In an aquatic environment, phospholipids spontaneously self-assemble into simple membranes. And this ability to form enclosed membranes of molecules is believed to be a critical step in the evolution of the first cells. Again, remember, compile your checkpoint questions, answer them, and email to me. In the origin of a cell, why would the formation of a simple lipid bilayer membrane not be sufficient? What else would we have to be part of such a membrane? So this is illustrating the initial laboratory experiment in which water-filled bubbles were made from phospholipids. Passive transport is a form of diffusion across a biological membrane. Whenever we use the word passive, it means that no energy investment, ATP molecules, chemical energy, molecular energy is not used. And the particles simply move from where they are more concentrated to where they are less. They kind of make space and spread out. So diffusion is the tendency of particles to spread out evenly in an available space. Diffusion across a cell membrane does not require energy. Because it does not require energy, it is called passive transport. Checkpoint question: why is diffusion across a membrane called passive transport? Some of you have completed at home diffusion experiments. So I will be sharing that with you all. I already watched Ryan's, and you've been doing well with your at home, and so this slide illustrates the process of diffusion. So we have a membrane separating this area where we have a solution, blue water, and the molecules of dye that are dissolved in the water, and just water on this side. So there's more water here, more solute and less solute here. So the solutes are going to move from where they are more concentrated to where they are less. So you have net diffusion of these orange particles is moving from where the left side to the right side where they are more. This is the semi-permeable membrane, and they continue to move, and eventually they will reach an equilibrium. At equilibrium, the concentration of these orange solutes on the left side will be equal to the concentration on the right side. At equilibrium, the molecules continue to move, but there is no net diffusion from one side to another anymore. So we can also illustrate this using two different solutes. So when you dissolve solutes in solution in solvent, you get a solution. So there's this orange solution and the purple solution. Orange solution is made of orange particles, and purple made of purple particles. And so there are more purple on this side, more orange on the left side. And so there will be net diffusion to the right of the orange particles, and net diffusion to the left of the purple particles. That process will continue until equilibrium is reached for both particles. They will continue to move, but at equilibrium, there will no longer be a net diffusion on either side. Midway in between, there was net diffusion of the purple from right to left, net diffusion of the orange from left to right. At equilibrium, the particles continue to randomly move, but there will be no net gain on either side. This would always be 3 particles and 3 particles, and this will always be 6 and 6 on these sides.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, can pass through the membrane unaided.
So osmosis is a specific or special type of diffusion that is relevant to water or only. So osmosis is diffusion of water across a membrane. So diffusion of water across a selectively permeable membrane is called osmosis. So when you take your beans and you soak them in water and they increase in size, it's as a result of osmosis going in. If a membrane permeable to water, but not to a solute, separates 2 solutions with different concentration of solute, water will cross the membrane, moving down its concentration gradient, moving downhill from where there's more water to where there's less. That process will continue of net flow of water from where there is more to less until the solute concentration on both sides are equal. So tendency is for solutions to want to equilibrate. So the checkpoint question is predict the net water movement between 2 solutions, a 0.5% sucrose solution and a 2% sucrose solution separated by membrane, not permeable to sucrose. So remember, compile the answers to your checkpoint questions and email them to me. So in this experiment, this is a U tube, and this is a selectively permeable membrane. Notice that you have 6 solutes on this side, and you have 13 solutes on this side. So the solution on the right side has a higher concentration of solutes than on the left side. So there's more water on this side. And this membrane is selectively permeable, but it is not permeable to the solutes in question. So the solutes cannot move across this membrane, so the water will flow because there's more water on this side. So that flow of water is osmosis, and water will continue to flow until to make the solute concentrations on this side and this side more equal. And so you see an increase in the volume of water on this side, the tendency of water to equilibrate, to try to keep the solution concentration uniform. So this is further illustrated. So these solid particles here are dissolved in water because water surrounds, forms a cluster around these polar molecules. And as they dissolve in it, as a result, there is more free water available on the left side and on the right side. And so the molecules will flow from where they are more to the right. And so the osmosis, because it's specific movement of water and not of the solid to the side.
The plasma membrane is permeable to water molecules. and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
So water balance between cells and their surroundings is crucial to an organism. Some of you have probably have found earthworms, and maybe if you drop salt on them, or slugs, and you draw salt on them, the salt draws water out of them and they shrivel and they break into pieces. So water balance is very important for cells, for their survival and functions. Toxicity is a term that describes the ability of a surrounding solution to cause a cell to gain or lose water. If you place a cell in an hypertonic solution, that cell will shrink. What is an hypertonic solution? It's a surrounding solution that has a higher concentration of solute than the inside of the cell, such as when you drop salt, which is very, very dry, highly concentrated in solutes. When you put it on a slug, on an earthworm, it draws water out of the cell and the cell shrinks. If you place a cell in an hypotonic solution, then the cell swells. So when you try to soak your beans in water, you notice an increase in size because the cell is in an hypotonic solution. It's just in pure water and it's taken in the water and it swells. In an isotonic solution, animal cells will remain normal because the solution inside and around the cell is the same, but plant cells will go flaccid because water cannot go in, and that influx of water into plant cells, that's in the hypertonic solution, is important for maintaining the turgidity of standing plants. So if we take if you take a blood sample from yourself and you wanted to observe it under the microscope, if you place that in water, which is an hypotonic solution, you will notice that your cells will soon swell and burst. If you place them in an hypotonic salty solution, they will shrink. But if you put in an isotonic solution, a salt solution, a saline solution similar to that of the plasma inside the cell, then you will see the robust, nicely shaped, donut-shaped red blood cells under the microscope. So checkpoint question: explain the function of contractile vacuole in freshwater paramecium in terms of what you have just learned about water balance in cells, and emphasize the function of the contractile vacuoles. So this is an animal cell, red blood cell. It lyses in hypotonic solution. It remains normal in isotonic solution, but it shrivels or shrinks in an hypertonic solution. Plant cells, the cell wall of plants kind of pushes back as hypotonic solution, water flows into the cell because the concentration of water inside the cell is less than the surrounding. So water flows into the cell, but then there's a pushback from the cell wall, allowing the plant to be turgid. So turgidity of plants is enhanced in hypotonic solution environment. If you place a plant cell in an isotonic solution, it would remain flaccid, or sometimes it might even it will go limp or just become flaccid. If you place them in an hypertonic solution, it will shrivel and you will see plasmolysis. During plasmolysis, the plasma membrane pulls away from the cell wall, and you can see that gap because the cell membrane is shrinking, and so you can see that moving away. This plasmolysis can also be observed in fungal cells and bacterial cells, or the other organisms that have cell membrane. So our knowledge of this plasmolysis is sometimes exploited, so that in bacteria, when we wanted to fight them with antibiotics, there are antibiotics that act on these differences, either blocking the formation of the cell wall of those organisms so that they are more susceptible to treat to antibiotics that are applied. So this is Chlamydomonas, just like a paramecium. They are organisms that live in pond water. Water flushes into them, and they have to pump those waters out using contractile vacuoles. Survival. This is showing paramecium actively pumping out water using its contractile vacuole. Otherwise, it will just fill up with water like a balloon and burst, like the red blood cell that we saw in that. But that doesn't happen because the paramecium has the contractile vacuole that's functioning to pump water rapidly out of the cell. This is illustrating plasmolysis in Elodia cells. So we place the cell in an hypertonic solution. So watch as the cell membrane moves in and the cell shrivels. So this is the cell wall. Look at the gap between the cell wall and the plasma membrane in this cell. You can observe it in onion cell, Elodia cells, plant cells, fungal cells, and some bacterial cells. So this is where the cell wall is for this segment. But look at how the plasma membrane, the interior of the cell, and the plasma membrane and the cytoplasm have gone in this time lapse micrograph showing plasmolysis. So plasmolysis occurs when you place plant cells or fungal cells or bacterial cells with cell wall in an hypertonic solution. So this is turgid Elodia cells growing normally and happily without plasmolysis. You can see cytoplasmic streaming. So there's the chloroplast organelles are moving around the wall. Certain smaller ions or molecules are moving between the cells. So you see how dynamic the cell is. When we draw the cell on the paper, it looks like nothing is going on, but this is a plant cell. So even inside the plant cell, there's movement. You remember the cytoskeleton guiding the movement inside the cells. And so the cells of multicellular organisms are that's inside the plant. Likewise, inside you and me, there's a lot of activity going on. So that is the working cell, it's alive. Transport proteins, they can facilitate diffusion across membranes, as we saw earlier on. Whereas hydrophobic substances can diffuse easily across the phospholipid bilayer through the fluid part of the membrane, polar and charged substances do not easily cross the membrane. And so these substances must be held to move across the membranes with the help of specific transport proteins called facilitated diffusion. So it is still diffusion because no ATP is utilized. So it does not require energy. So it still is diffusion, but it is facilitated by the transport proteins. It's also diffusion because it's movement down a concentration gradient. The substances are moved from where they are more to where they are less, and no energy is used. But because the hydrophobic membrane presents a barrier, the proteins have to facilitate the movement of these molecules across the membrane. So you have co-transportation of sodium and glucose, for instance. Transport transport proteins, they are also helpful in the rapid diffusion of water in and out of certain cells. For example, in the kidneys, in the body, there's recycling in the human body, recycling of water, thanks to the aquaporin, which is very, very abundant in kidney cells. So this protein channel aquaporin was discovered by Peter Algre, who received the Nobel Prize in 2003 for this discovery. These are protein channels that specifically allow influx of water. So hence they are just aquaporin water pores. Checkpoint question: how do transport proteins contribute to membrane selective permeability? The transport proteins only move specific substances. So aquaporin is specific for water, for instance. So this is a transport protein. Specific molecules move across it. So even the membrane proteins are very, very specific in what they move. So the entire plasma membrane remains selectively permeable. Doesn't allow everything to go through. Dr. Peter Agre received the 2003 Nobel Prize in Chemistry for his discovery of aquaporins. His research was on the rhesus protein used in blood typing, and in studying this protein, he discovered another smaller protein, and he investigated further, and that investigation led to the discovery of aquaporins. So in the experiment, they had used frog eggs to test the function of this unknown protein. Frog eggs are known to be impermeable to water, but he found that whenever they took the smaller protein and the RNA for the smaller protein was inserted into the protein frogs, and then within 72 hours, the frog eggs were able to rapidly take in water, but the control cells were not. So they also compared the smaller protein with the RH protein, and they found that the sequence has no resemblance. So they realized immediately that this was a separate and different protein than the RH protein. And Peter Agre's colleagues, this is one of the areas where you remember the scientific process, that the scientist works with others in the community and they communicate with other scientists. And through that communication, Dr. Agre found out that scientists have been trying to identify the factor, the protein, or the molecule that's responsible for the rapid influx of water into cells. So these are the results of the experiment, the controlled eggs, which were not injected the RNA for making the aquaporins. The relative volume remains low. Now, within look at the rapid increase within 72 hours. The cells filled up with water and ruptured. The eggs that were injected with the RNA. So, cells expend energy in active transport of a solid. So we've looked at transport across membrane. We have discussed diffusion, which is a form of passive transport. Passive transport does not use ATP. Active transport uses ATP. So in active transport, a cell must expend energy to move a solid against its concentration gradient. So in comparing active transport with passive transport, passive transport tends to involve movement of particles from where they are more concentrated to where they are less, which is downhill transport. Active transport tends to move materials uphill, which requires a push and energy from where they are less concentrated to where they are more. So the energy molecule of ATP is the functional energy that cells use the energy currency of the cell, it supplies the energy for active transport in biological cells. So remember your checkpoint questions, answer them, and turn them in. So this is illustrating active transport. So we have a transport protein and a solute to be transported against its concentration gradient binds to the active sites of the protein. And the protein is energized by ATP through a phosphorylation process in which ATP gives the phosphate group and the ADP remains in the system. As a result of the phosphorylation of the protein changes shape and allowing it to be able to export the solute to the other side of the membrane. As soon as the solute is ejected, the protein returns to its original shape and is ready to bind more solute and continue the process of pumping out unwanted molecules from regions where they are less concentrated to where they are more concentrated, depending on the needs of the cell. So let's edit this video.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here's how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, adding energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
So exocytosis and endocytosis, they help transport large molecules across the membranes. So there are two mechanisms that these are two mechanisms that the cell uses to move molecules across the membrane. Exo from the word exit and cyto of the cell. So exiting, moving large molecules out of the cell. Exocytosis is used to transport bulky molecules such as proteins or polysaccharides from the inside to the outside of the cell. Endocytosis, endo, inward coming, is used to take in large molecules into the cell. So in both cases, materials are transported in packaged in vesicles, and these vesicles of biological membrane fuse with the other biological membrane. So taking into cell. So endocytosis, there are two kinds of endocytosis. In phagocytosis or cell eating, there's engulfment of a particle in the, by a cell, wrapping its cell membrane around it, forming a vacuole. Proteins such as amoeba and those smaller organisms feed in this manner. And so also some of the white blood cells in our body, they're able to engulf bacterial infection this way and help to put them in a vacuole and digest them and protect us in that manner. The second kind of endocytosis is receptor-mediated endocytosis, which uses membrane receptors for specific solutes. So, and takes them in. So checkpoint question, as the cell grows, its plasma membrane expands. Does this involve endocytosis or exocytosis? Explain. So this slide illustrates phagocytosis and receptor-mediated endocytosis, the two types of endocytosis taken in. So this is the extracellular fluid, the the cell here forms a false feet or pseudopodium by extending its cell membrane and wrapping its plasma membrane around the food, budding off a food vacuole into the cytoplasm. Now, lysosomes inside the cytoplasm can fuse with this food vacuole and digest the nutrients, and the chemical elements will be returned into the cytoplasm. So kind of recycling taking place, or feeding, cell eating. In receptor-mediated endocytosis, there are receptors on the membrane surface. It recognizes its specific molecules that it's responsible for transporting and binds to them. Then typically there's protein coats on the inside. So it forms parts of a coated vesicle into the interior of the cell. So endocytosis, it's taking in specific molecules here. Endocytosis, phagocytosis type is taking in food particles.
Cytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure. Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
So this is how insulin, for instance, is secreted. This is how trypsin and those different enzymes are secreted.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
So pinocytosis is very, very important in us getting the nutrients from digestion products into all over the body.
In endocytosis, membranes invaginate or pinch in to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded. Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
And let's look at energy and the cell. So cells transform energy and matter as they perform work. Initially, we established the fact that all of the energy that we use in our planet comes from the sun. There are basic ways in which energy is available or functioning in life. Kinetic energy is the energy of motion, the energy that's doing work, that's making things move, shake, boil, and all of that. Potential energy is stored energy. It's energy due to the location or structure of matter, and it includes chemical energy. The energy that binds the carbon, the atoms of carbon to hydrogen and oxygen in glucose, for instance, is a form of potential energy. When you put a ball uphill, it has energy due to its position, and it can roll downhill when you drop it using that potential energy. The stored energy in food, in potatoes that you feed, is also a form of potential energy, and so on and so forth. After studying the transformation of energy, scientists came up with the laws of thermodynamics. And the laws of thermodynamics states that energy can change form, but cannot be created nor destroyed. So the only energy that we have is harvested, obtained from the sun. We cannot create energy or destroy energy. But energy can be transformed. Energy transfers or transformations in, however, increase the disorder or entropy, which some energy being lost as heat. So when you have a car leaving a spot, sometimes going very, very rapidly, you can sometimes see heat almost burning coming out of the tire. When you are running, your body temperature goes up, you perspire more, because some of the energy that you are transferring from your food to the kinetic energy of motion that you are running is being converted to heat, warming you up. Energy during transformation of energy, whatever energy becomes disordered, becomes heat, is referred as entropy. It's released to the environment and will not be back in any orderly form that we can utilize to do work. Checkpoint question: how does the second law of thermodynamics explain the diffusion of a solid across a membrane? So this is comparing energy conversion in a living thing to a non-living thing. So similar to your automobile, gasoline, when you fill your car with gasoline, that gasoline will burn with oxygen to release the energy that was locked up in the chemical molecules of gasoline in the hydrocarbons. As the hydrocarbons give kinetic energy of movement to the car moves forward. Some of that energy is converted to heat energy. You can feel, you have to cool your engine or it would explode. And you can even feel your heat on the tires and so on and so forth. And you see the exhaust releasing carbon dioxide and water to the environment. Compared to the living cell uses glucose, which instead of gasoline, and but it bonds with oxygen, giving off heat during cellular respiration, converting the energy of glucose to ATP, allowing the cell to work, whether you're running, you're walking, you're cooking, or whatever you're doing. So ATP enables you to do it. And the byproduct, the waste products from that cellular respiration, is similar to the automobile waste products of carbon dioxide and water. However, the efficiency of the energy conversion in a car is only about 25%. In a living cell, it's 34% efficiency. So the living cell is more efficient at energy conversion than the automobile. But either way, still a lot of energy that is not properly converted. 66% goes to entropy here, 75% goes to entropy in this system. So let's watch this video on energy concepts.
Energy is the capacity to cause change. Kinetic energy is the energy associated with motion. Energy that is not kinetic is called potential energy, and is energy that matter possesses because of its location or structure. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy.
The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow. When the arrow hits the target, its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let's rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, some of the energy can be used to do work, but some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. As the arrow flies through the air, heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Energy is a very disordered kind of energy. It has the highest amount of entropy, or disorder, of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. The body breaks down food molecules. The stored potential energy from food can be converted to kinetic energy. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP. Water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work, such as pulling back a bowstring, but the heat energy that is generated cannot be used to do work.
So chemical reactions take place constantly in the body as matter is exchanged and energy is generated and transformed in the body. So chemical reactions taking place in the body can either release or store energy. So we define energies as a chemical reaction as exergonic, exciting if it releases energy, and as endergonic if it requires energy and yield products that are rich in potential energy. So the products of exergonic reaction would have less energy than the reactant. The products of endergonic reaction would have more energy than the reactants. We use the term metabolism to describe all of the cell's chemical reactions. So metabolism encompasses all of the cell's chemical reactions. Checkpoint questions, remember to compile, answer, and email them. Remembering that energy must be conserved, what do you think becomes of the energy extracted from food during cellular respiration? So reactants yield products. This is an example of an endergonic reaction. Exergonic reaction. This is a higher level of energy in the reactants than in the product. So the reaction gives off energy. Energy is exiting the system, exergonic reaction. The difference in the energy of products and that of the reactants is the amount of energy that's released from the system. This is an example of an endergonic. Reactants have less energy than products, so energy input during the reaction makes the products have more energy. This is the amount of energy required for the reaction to take place, and the product will have more energy than the reactants in an endergonic reaction. In cells, ATP drives cellular work. In biological cells, exergonic reactions are often coupled with endergonic reaction, in which one reactant is giving off energy and another reaction is taking in energy. So ATP powers nearly all forms of cellular work, and it does this by phosphorylation. The transfer of a phosphate group from ATP is used. It is involved in the chemical, transport, and mechanical work. So all of the forms of work that take place in the cell, chemical work, transport work, and mechanical work, involves the phosphorylation by ATP. So the ATP molecule is recycled in the cell. Checkpoint question, explain how ATP transfers energy from hexagonic to endergonic processes in the cell. So ATP, adenosine triphosphate, adenosine with 1, 2, 3 phosphate groups attached to it. These are high energy bonds that join the phosphate group to the adenosine nucleoside. By hydration, adenosine triphosphate is converted to adenosine diphosphate, and the phosphate goes on to bind to another molecule in a phosphorylation, and the bond between the phosphate number 2 and phosphate number 3 is broken. The energy of that bond is released. So to do chemical work, ATP gives the phosphate group to phosphorylate the reactants, making the product or formation possible. As soon as the product is formed, the phosphate groups is released again into the cytoplasm, and there's always a pool of ADP, adenosine diphosphate, and phosphates that's there in the cytoplasm. So the ATP, in doing chemical work, at the end of that chemical work, it will facilitate the reaction, which is the actual metabolism, but ATP itself will be changed to ADP and inorganic phosphate. So the phosphorylation, taking the phosphate from ATP here, powers the reaction. Once the product is formed, the phosphate returns. And when the phosphate is moved from ATP, it becomes ADP. During transport work, again, as we saw earlier on, it can bind to the transport protein, causing a conformational change in protein structure, facilitating transport across the, enabling transport across the biological membrane, again, ADP and PID. During mechanical work, as in the muscle contraction, so ATP releases its phosphate again to phosphorylate the motor proteins and facilitate the sliding action. And then at the end of the day, ADP and inorganic phosphate will be formed. So protein filaments move and contraction takes place. So like I said, the ATP involvement in mechanical work, in transport work, and chemical work in the body is kind of a process that involves recycling of ATP, ADP, and inorganic phosphate. ATP, by hydrolysis, gives up its phosphate group, provides the energy needed to do cellular work, and eventually the inorganic phosphate and ADP are released into the cytoplasm. During the process of cellular respiration, there is new energy that is being converted, and it's again used to bind ADP to inorganic phosphate to synthesize ATP again. And this cycle will continuously take place in the cell. So the cell is always making ATP, using it, breaking it down to ADP and inorganic phosphate. During cellular respiration, rebuilding the ATP in the cycle of life. Finally, let's look at how enzymes function. So what are enzymes? Enzymes are protein catalysts. They speed up the cell's chemical reactions, and they do so by lowering the activation energy. So these enzymes are protein catalysts that decrease the activation energy needed to begin a reaction in the cell. So this is a reaction in the cell. The black line represents how the reaction would have taken place, how much energy would have been needed for the reaction to take place without an enzyme. And the red graph here, the red line here shows the reaction in the presence of enzymes. So this is the activation energy of the reaction. It's greatly reduced from activation energy of A to B. There's a great reduction, causing the reaction to take place much more rapidly speeding up the rate of reaction. So reactant, this is the without enzyme, this is the activation energy needed. With the enzyme, this activation energy. Typically, enzymes have an active site that feeds the substrate. When the enzyme is fed into the enzyme's active site, when the reactants fit into the enzyme active site, it is more convenient for the reaction to take place either by repositioning or impacting the structure of the substrate, so such that it can be separated into two, or it can be brought into contact with another reactant. In enzymatic reactions, the reactants are actually called substrates. And, so, checkpoint question. The graph below illustrates a reaction with and without an enzyme. Which curve represents the enzyme catalyzed reaction? What do lines A, B, and C represent? Remember to compile your checkpoint questions and email to me. Now let's look at how enzymes work.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. Enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
So specific enzymes catalyze cellular reaction. So one important property of enzymes is that the enzymes are very specific in their actions. And enzyme substrates fit specifically in its active site. So this is why it's easy to name enzymes according to the substrates that they act on. So sucrase acts on sucrose, lactase acts on lactose, and so on and so forth. The following figure illustrates the catalytic cycle of an enzyme. So the enzyme is available with an empty active site. This is sucrase in this instance. Substrate sucrose comes in, it fits snugly into the active site. If lactose comes in, it will not complete the reaction. Then there's hydrolysis, and the products fructose and glucose are released. The enzyme remains unchanged at the end of the reaction and is ready to pick up another sucrose and continue very, very rapidly, speeding up the rate of reaction. Checkpoint question, explain how an enzyme speeds up a specific reaction. Enzymes inhibitors are also present in the cell, and they are important because they help to regulate enzyme activity in the cell. And enzyme inhibition take place by competition. So we have a competitive inhibitor can compete with the substrate for the active site of the enzyme. When the inhibitor blocks the active site of the enzyme, the site is not available for the normal substrate and the reaction will not proceed. you can overpower the competitive inhibitor by providing more of the substrate of the regular substrate. A non-competitive inhibitor alters an enzyme's function by changing its shape. So it binds to the enzyme in a location different than the active site, causing the shape to change and so that it no longer fits its normal substrate. And allosteric enzymes might work this way. They might pull away from the site when the enzyme is needed and then bind to the enzyme again when it's not. So these are the regulatory processes within the cell to keep the enzymes from going haywire. Enzymes can also be regulated by feedback inhibition, which helps to regulate metabolism. When a particular product reaches the maximum concentration, it can act as a feedback to block further production of a particular intermediate so that there's not an excess production of one substrate and utilization. There's not excess production of a product or exhaustion of a substrate. So this is the active site for the enzyme. Normal substrates fit. If there's competitive inhibitor, it might fit the site of the enzyme, but by occupying that site, it blocks the normal substrate, and that reaction, that metabolic process will be blocked until the competitive inhibitor leaves the active site of the enzyme. And it can be the non-competitive inhibitor binds to a different site and changes the shape, and the shape no longer fits the normal substrates, and so the reaction is placed on hold until somehow by other molecular attraction the non-competitive inhibitor is dislodged from the enzyme. In feedback, product D acts as an inhibitor of the step 1, reaction 1 of the enzyme. And so whenever this is present to maximum, then this enzyme 1 is inhibited and the product will not be produced. When the product depletes again, then enzyme 1 will be freed up to function on there. So checkpoint question, explain the advantage of feedback inhibition to a cell. Many beneficial drugs act as enzyme inhibitors. Ibuprofen, for instance, it inhibits prostaglandins so that you don't feel pain, you don't have inflammation. It doesn't stop the cause or the foundation, but you just don't feel the effects of it, such as the pain and the inflammation. Enzyme inhibitors have also been developed as pesticides and deadly poisons of chemical warfare. So they can, if a nerve gas, for instance, would inhibit important enzymes in the metabolic pathway, and within a very, very short time, the entire system shuts down, because enzymes are needed for all of the activities in the cell, especially cellular respiration, all of those things. Checkpoint question, what determines whether enzyme inhibition is reversible or irreversible? So upon this from this chapter, from this lecture, and your studying of this chapter, you should be able to describe the fluid mosaic structure of a cell membrane, the diverse functions of this membrane proteins. Should be able to relate structure of the phospholipid molecule to the structure and properties of the cell membrane, to define diffusion and describe the process of passive transport, Explain how osmosis can be defined as the diffusion of water across the membrane. Distinguish between hypertonic, hypotonic, and isotonic solutions. Explain how transport proteins facilitate diffusion. Distinguish between exocytosis, endocytosis, phagocytosis, pinocytosis, and receptor-mediated endocytosis. Define and compare kinetic energy, potential, chemical, and heat energy. Define the two laws of thermodynamics. Explain how they relate to biological systems. Define and compare endergonic and exergonic reactions. Explain how cells use cellular respiration and energy coupling to survive. Explain how ATP functions as an energy shuttle, how enzymes speed up chemical reactions, how competitive and non-competitive inhibitors alter enzymes activity. Explain how certain drugs, pesticides, and poisons can affect enzymes. Thank you.